few days of lecture left. Including the day, I think 6 lectures left. Yes, 6 lectures left. So, semester is winding up. A few announcements today before we get started. Don't forget about DNA Day on Monday. You can check out the schedule a bit, see if there's anything you're interested in. It's happening at the campus center. Hopefully you have gained an appreciation of DNA in this class and are interested in it in some capacity for your major. So you can check out DNA Day. Higher celebration for DNA. Announcements about the exam. Remember that so you're getting them back in recitation today if you didn't get them back yesterday. If you have any questions about the grading, come and talk to me. You can get point adjustments, or at least present your case for point adjustments to me until next Friday. After that, I won't accept any more adjustments. After that, you are always welcome to come and talk to me about concepts if you didn't understand something. But you have until Friday to talk to me if you think that something is weird about the grading. Okay? The only thing that your TIs can adjust for this exam is if there are errors in the addition of your points. Everything else you have to come to me for. Are there any questions about the exam at this point? We'll talk about the final a little bit next week. We'll start talking about how you can prepare for that. All right, if you haven't looked yet, the final for this class is on Friday of finals week, so the very last day, early in the morning, 7:30 I think it starts. Why they would do that to you, I don't know, but that is the time we have been assigned. So 7:30, the last Friday of finals week, Friday the 13th, very auspicious date. So make sure that you keep track of that. I will be giving you study guides and mastering biology and all of that. We'll talk about that next week though. Okay? Are there any questions at this point about the exam or the final? You will have homework this weekend. I'll post that when I get back to my office today. So keep an eye out for that. Any other questions before we get started? Thank you. So the questions about the average, everyone settle down. So the questions about the average for this exam, I don't have it yet because the TIs from today are still getting grades entered. I will, as before this evening, I will try to get all of the grades updated through this exam. So any homeworks and quizzes and things that aren't yet posted on Malima, I'll try to get this posted today. And then I'll be able to give you an average then. I'll make an announcement. But as always, please double check your scores carefully to make sure we haven't made a mistake. I very much want to make sure we get you the points that you deserve. So if something looks weird on Malima and we have something recorded, let us know. Okay? Either the TI or come to me and we'll get that fixed up. Okay? All right. So today we're going to talk about signal transduction. If I can shout at you over the noise of whatever gardening is going on outside. So we're going to start off with, for this last section, we're going to talk about signaling. So we're working our way up. We're going to start to put together now components that we've talked about throughout the rest of the course. So we're going to be looking at protein interactions. We're going to start off with neuronal signaling. So we're going to be revisiting those membrane transport proteins. Hopefully you haven't forgotten about those. Those are very important in this first sort of signal transduction system we're going to be talking about. We talked about briefly on Wednesday the idea of communication and distance and how the way you communicate changes over different distances. We talked about that relative to two people communicating. And I wanted you to think about how cells communicate, particularly relative to distance apart. So this is where we ended up. So we're going to fill these in now. You had a few seconds at the end of the last lecture to look at it. Let's go over them. So cell-cell contact. What do you think the distance that happens over? Touching.
Is this a chart?
Autocrine.
Oh.
What kind of signaling is that? Self signaling. Auto self. Endocrine system. What's the distance? You can guess this long, short. Endocrine is a long distance signaling system. So signaling cells that are fairly distant from where you are, other parts of the body. What about neuronal or synaptic? So this is where we're going to start. Neurons and their synapses and how that works. And then what does that leave for paracrine system? Short distance, signaling cells in your sort of immediate area. So biological systems have different modes of signaling depending on who they're trying to communicate with. And we're going to look at a number of those for these last few lectures. All right, so we talked about, if you want to keep track of how cells are signaling each other, what are those signals, how are they detected, over what distance are they sent. We're going to talk specifically about two types of cellular signals. And those are electrical signals, which play an important role in neurons and signal transmission, and then chemical signals, molecules that cells can send out to send information to other cells. All right, so we're going to start with neuronal signaling, then we'll move on to GPCR, which is G protein-coupled receptor signaling, and enzyme-linked receptor signaling. So we're going to start with neuronal signaling, that sort of very close interaction, then GPCR and enzyme-linked over longer distances. So we're going to spend the next few lectures looking at neuronal signaling. So the electrical signals in nerve cells and how those are generated, how they are propagated across the cell, and then what that results in sending the signal to the next cell in that nerve pathway. And so we're really going to get down into the details about how these electrical signals are generated. So you're going to need to know action potentials inside and out. You're going to need to know what channels are involved, what ions, and how each of those plays a role in generating that action potential. All right, so let's start off with, if you wanted to hit your professor, I'm not suggesting you do that, but if you wanted to, what are the nerve impulses? Somebody just tried. So if you wanted to hit your professor, what are the nerve impulses, right? So you had to have that thought. Then where does that signal get sent, right? How does that translate into movement? All right, let's look at the particular example of, so if you've been to the doctor and they check your reflexes, right, they tap your knee, and you have, you try to kick the doctor. So kick the doctor rather than me. So we're looking specifically at that knee area, right? And how does that reflex work? So similar pathway. We're talking about wiring diagrams for neurons. So we looked at that pathway. There are a number of different neurons involved. So we're going to talk today about the types of neurons and how they're organized and their different structures. And then we'll get into ion channels and ions, and then we'll look at how they generate action potentials. So the doctor hits your knee. It's stretching a tendon and stretching sensory receptors. right? Those receptors then send a signal to a sensory neuron. It goes into your central nervous system. It communicates with interneurons and motor neurons that send a signal back to your muscle that says we've received the signal. Go ahead and do something. Kick. The reason that this works is that this sensory pathway doesn't really go through your sensory nervous system. It doesn't need any higher brain processing, right? It's wired that if these stretch sensors are activated, then contract and relax your muscles in such a way that you kick your leg. So the pathway, you get a stimulus. Stimulates sensory neurons, those receptors. If the information is integrated through interneurons, so interneurons are neurons that integrate information from one neuron and send it out to another. That information gets sent to motor neurons. Those motor neurons send a signal out to the effectors, in this case, muscles. And that signal in those effectors then generates a response, in this case, kicking your leg. So we can simplify that pathway. Excuse me. So we have our muscle and our sensory neurons. They send a signal to our spinal column, where it integrates with the motor neuron. So they send that signal to the motor neuron, which goes back and sends the signal to the muscle. All right, so let's, all of these impulses, right? We receive a stimulus, it's integrated, and then there's some sort of output. All of these are due to different kinds of neurons. So we've already mentioned two, sensory neurons and motor neurons. So let's look a little bit at neuron structure. That's going to be important to understanding how these electrical signals are propagated in neuron cells. So in our nervous system, sending these signals, there's two sorts of cell types to be familiar with. The neuron cells themselves, which we spent a lot of time talking about. These are the excitable cells. So they receive and transmit stimuli. And then the glia cells, which serve as support cells for those neurons. So they support and protect the neuron cells. But first, let's look at the neuron cells and the components of the neurons. We look at a neuron cell, composed of a couple of different parts. It's got dendrites, so these projections off the cell body. It has sort of a long, skinny projection off that cell body, we call the axon. And it's also got on the other end projections that are called synaptic termini, or terminals. All right, so the incoming signals are picked up by dendrites. The signal accumulates in the cell body. There's a trigger zone right at that cell body axon interface. It's called the axon hillock, so where cell body turns into axon. And then the signal is sent down the axon to the synaptic terminals, where neurotransmitters are released to send the signal on to the next cell. So those dendrites again, they're picking up signals from outside the cell, and that signal is converted in the cell to a flow of ions, the current. So in neurons, we are creating current by moving around ions. Think about how we can move ions around. The trigger, right, once enough signal has been accumulated to pass a threshold, that trigger transforms that input signal at that axon hillock into action potentials. So changes in charge across the membrane that propagate along the axon and can be propagated long distances. So we send the signal from the dendrites down the axon to the synaptic terminals. And then an action potential arriving at that synaptic terminal causes the release of the neurotransmitter. That's the output signal, and it's picked up by the next neuron in the circuit. So the neurons are the ones that are moving ions, creating current, creating voltage, and propagating that to the next cell. That axon allows the cell to propagate that information over long distances, these long skinny projections, relative to a cell anyway. And then we come back to those glial cells that support and protect that neuron. Right? Support and protect, you have a long skinny projection from a cell, that a current is being sent down. Right? So there are several different types of glial cells that help support that structure. And then you see in blue here, these glial cells, they wrap a myelin sheath around that axon, which helps insulate that axon. Right? We're talking about changes in current, moving ions around. By insulating it, it's helping that information travel faster, protecting it from the surrounding environment. So these neurons, probably one of the most extreme examples of a neuron are the giant axons and the stellar nerves of squids. So these axons can be as large as 1 millimeter in diameter. That's really big for a cell, right? And they project along the length of that animal. All right, these are rapid response sort of reflex neurons. If you ever want to do recordings from a nerve cell, a 1 millimeter axon is a really big thing to aim for with your physiology equipment. So that's why people like squids for physiology. Nice big nerves to record from. All right, so these neurons can be really large, the axon projections, and they can travel over really large distances relative to those cells. There are, depending on where the neuron is, what its job is, what tissue it's in, what circuitry as far as sending and receiving signals. Those neurons can take on lots of different forms relative to the dendrite and synaptic terminal arborization, so how branched it is. It can also vary relative to the placement of the cell body and the dendrite and the axon. So there's lots of variation in neuron form and function. There are I think there are over 10,000 specific types of neurons that have been described from the human brain. So there's a lot of variation in how these neurons are arranged to detect and propagate signals. There are 3 general classes I want you to be familiar with. Those are bipolar neurons, unipolar neurons, and multipolar. Polar being how many projections come off that cell body. So in bipolar, we have our cell body. We have a projection off of each end, 2 projections, bipolar. Unipolar, we have a short projection, a single short projection off of that cell body that then branches to form those dendrites and axons. And then multipolar, multiple projections off of that cell body to achieve dendrites, axon, synaptic terminal. All right, so there are lots of variations, as I said, in the branching and the arrangement, but these are the three general classes of neurons that you're going to see. All right, and they vary based on where the dendrites are, the axon is in the synaptic terminal. So that's multipolar, bipolar, dendrites on one end, axon and synaptic terminal on the other. Unipolar, so we have dendrites, super long axon, and then synaptic terminal. So those are structural classifications of our neurons. And they are linked with functional classification. So most multipolar neurons, right, are in the central nervous system and serve as motor neurons. Bipolar neurons serve as sensory neurons in the eyes and nose, and are often used, that structure is used in interneurons, for neurons that are connecting to other neurons. And then unipolar, all the rest of the sensory neurons that aren't in your eyes and nose are typically unipolar. All right, so those are structural classifications that correlate roughly with functional classifications. We look at those functional classifications and give you a little bit better definition of, I've already mentioned sensory neurons, motor neurons, interneurons. Let's give you some definitions for those. All right, sensory neurons, afferent action. So they're sending their potentials towards the central nervous system. They're picking up some signal on the periphery of the nervous system, and they're sending that back to the central nervous system. Right? They're saying, I've picked up some information, do something with it. Motor neurons, efferent action potential. So these are neurons where the potential is being, and the signal is being sent away from the central nervous system. So the central nervous system has picked up some signal from the sensory neurons, and it's sending out a command for some sort of action. So interneurons, these are association neurons, so that's why they're typically in that central neuron system. They are transmitting from one neuron to another. They're sensory, sending signals to central nervous system, interneurons integrating across neurons, and then the output is back out motor neurons. If you wanted to design a neural circuit from the periphery to the central nervous system back out into some action, you would need all of these types of neurons, like we saw in the knee-jerk reflex. But regardless of structural classification or functional classification, neurons in general are cells that are excitable. So when I say a cell is excitable, what do I mean? Gets really worked up and it gets excited about molecular biology. All right, what does it mean when a cell is excitable? Membrane potential. What do we mean by membrane potential? We're setting up what across a membrane? Concentration gradient of, I think I heard it, ions. So charge. So we're setting up gradients across the membrane. We're stacking up charge on one side or the other. And then how do we generate the electrochemical signal once we've set up that gradient and that charge differential? What is a current?
Flow of electrons.
The flow of ions. So what do we need to get those ions across the membrane? Our channel proteins, our transport proteins. So electrical excitability means that the cell is moving ions across the membrane. So we need some sort of transport proteins. We're going to talk both about channel proteins and active transport carrier proteins. relative to neurons and the electrochemical gradients of ions that are set up to propagate this electrical signal down the cell, down the axon. We're going to pay attention to sodium concentrations and potassium concentrations. So those are the two ions we need to move back and forth across the membrane. So think about, when we talked about membrane transport, what kinds of transport proteins involve sodium, what kind of transport proteins involve potassium, what kind of transport proteins involve both. All right, we're going to see those playing a role in action potential. All right, ion channels. Quick review from membrane transport. All right, so this is the generalized ion channel that we looked at. How are things going to move? How is that solute going to move?
Oh, I just grabbed it off your desk because I thought it was I know. Me. I think it's never. I need to eat.
They can pick E.
It's E.
Oh, like, here's the picture. Generalized ion channel. The question is more general than just the picture, right? All right, everyone get their answers in.
Mom.
Let's take a look.
You say mom. You say mom. We just started.
Ion channels flow when the channel is open. Pretty straightforward. But that means relative to controlling that membrane potential, the cell can control that membrane potential and the current generated by opening and closing different ion channels. All right, so let's look at a couple of different ways that ion channels can be opened and closed. We looked just generally at them and how they function when we talked about them as membrane transport, but there are different ways that a cell can control what state that channel is in. All right, so the first one, well, right, just a generalized conformational change, right? In some state it's open, in some state it's closed. That channel is blocked. And we want to talk specifically about how the cell is controlling whether or not that channel is open. We talked already a little bit about localized conformational change. So you can think of these as having a gate, some part of the protein channel. So some small part of it, rather than the entire thing changing conformation, some small part of it is opening and closing. So localized conformational change. You're going to see in action potential a different kind of membrane transport protein. One that has both a gate on it and the ball and chain stopper. So different ways to close that channel depending on the state of the membrane. So we can have it open and activated. We can close the gate and deactivate it. Or under certain physiological conditions, a different part of the protein can plug the hole. All right, some part of the protein that acts as a blocking particle, a drain stopper really. All right, so we've got a gate and a drain stopper that are going to function, block that channel under different conditions. And we're going to look at that relative to action potential. All right, if the protein has multiple ways to block off that channel, how can it block off ion flow? I mean by closed? What do I mean by inactivated? All right, everyone get their answers in? Take a look.
I don't know.
All right, so when the protein or the channel has multiple ways to plug that opening, right? If it's got a gate to open or close, if it's got some sort of blocking particle, we call that activated or inactivated. So one or the other or both can stop ions from flowing across that membrane. What did I say? Stop. Okay. Okay. So ion channels, remember they're specific to a particular type of ion. So when sodium channels open up, potassium ions are not going to rush through. And different sorts of stimuli control the opening and closing of different types of ion channels. So by opening and closing channels at different times, this is how the cell is going to control the membrane potential and the current, the voltage across that membrane to propagate that signal. There are a few other ways that we can open up ion channels you should be aware of. So there are ligand-gated ion channels. So this is where the channel needs to bind some stimulus, some signal, in order to open up. So once that signal is bound, the gate opens. Without that signal, the gate remains closed. There are voltage-gated ion channels. We're looking at the way that charges are distributed across that membrane. So in one particular instance, we have more positive ions on the outside, negative on the inside. That gate is closed. If that membrane potential reverses, that triggers that channel to open. So that's another way that cells can control when these ion channels are open or closed. All right, one example of this are voltage-gated potassium channels, which are mitten-shaped proteins, and that thumb paddle shape changes position, right? So it swings from down here to close the potassium channel to up here to open it, depending on the voltage across that membrane, right? Because that thumb is charged. So setting up that differential potential across the membrane changes where that thumb, the position of that thumb in the protein, opens and closes the channel. And then the final sort of way that we can open up ion channels that we're going to talk about anyway are stretch-gated ion channels. So think about the example of that knee reflex. So we hit the knee, we stretch that tendon. How does that send a signal? One potential way are these stretch-gated ion channels, right? They are normally closed, and through physical stretching of the membrane, you can pull open those channels. So it's more of a physical control than a chemical or an electrical control. Oh, there was one more. I don't have a picture for this one. You can also control ion channels by the state, their phosphorylation state, so whether they have extra phosphates added or removed. So there are all sorts of ways that ion channels can be controlled relative to when they are open and when they are closed. We're going to talk about some specific examples of these different ways, different types of ion channels relative to electrical signals in neurons. talking specifically about neurons and setting up that potential, that electrochemical gradient across the membrane. I've already said that we want to pay attention to sodium and potassium. Where do you think the concentration, or how do you think the concentration gradients are set up for these two ions relative to a neuron? So inside the neuron, relative to the outside of the cell, what are the concentrations of these ions? Think about our channels. Think about, in particular, we talked about an active transport protein for sodium and potassium. How does that work? Which way are things shuttled? How many of them are shuttled? What would that mean relative to the inside of a cell?
Yeah, that's why I'm saying it's before that, because I thought it was C, but then it said inside. So that's
Is it not working? Oh, that's just... We'll get there in a minute. All right, does everyone have a chance to think through that? Let's take a look. All right, so general consensus is that the answer is D, low sodium and high potassium. Why? Justify your answer.
That's how the heat pump works.
That's how the what pump works?
Heat pump works.
That's how that sodium potassium pump works? How does the sodium potassium pump work? Three sodium, 2 potassium, in what direction? Sodium in, potassium out?
Sodium out.
Sodium out, potassium in. Which way would generate answer D?
sodium and
Which way is going to give us low sodium inside the cell and high potassium inside the cell? Pump sodium out, pump potassium in. All right? So D is the right answer. Inside the cell, we have low sodium and high potassium. All right, this is going to set up the electrochemical gradient the neurons are going to use to generate those action potentials. So if we look at the concentrations, so ion concentrations inside and outside neurons and their axons for sodium, potassium, and chloride. So outside, so your squid axons, mammal neurons, the cat. So outside concentrations in millimolar for sodium and chloride, salt, NaCl. Inside for potassium. So there's a difference in ion concentration inside the cell relative to these ions and outside the cell. So we've already talked about this. What is doing this? What's setting up this difference in concentration? Our sodium-potassium pump. It's chugging away, sending 3 sodium out for every 2 potassium it brings in. So it's setting up that differential in concentration. So if we have this difference in concentration for sodium and potassium, do we have a voltage difference yet? Is there a difference in charge between the 2 points that we're measuring in our electrical field?
No.
Justify your answer. What do we need to set up a current?
More different charges on both sides.
We have to have different charges on both sides, and what do we need to do with those charges? They need to move. All right, we need that ion flow to generate our voltage. So once we set up our concentration gradient, our sodium-potassium pump pumping away, right? We need one more component. We need those ion channels to open up for those ions to flow across. So let's look at, now we've set up our membrane potential, all right, with differences in concentration in sodium and potassium on either side of the membrane. What happens when we open up ion channels for potassium? And what happens when we open up ion channels for sodium relative to the membrane potential? All right, so let's start with potassium. I think we have just a few minutes left. All right, we looked at our system, our concentration gradients that we have set up by our sodium-potassium pump, and we put in a potassium ion channel. When we open up this ion channel, what direction are the potassium ions going to flow?
Go back.
Yep. Think again. What direction are the ion channels going to go?
Which one's going to
Think about where the concentration gradient is. Alright, potassium, we've set up our gradient, we've opened up our ion channel. Where are those potassium ions going to move? Now everyone get their answer in? Let's take a look. It's going to move from inside the cell to outside the cell, because inside the cell is where the concentration gradient is. Well, in there today for Monday, think about what that means for the current and the voltage we're measuring. Think about what that's going to do to our membrane potential.